I guess it's 28. Could do stand-up routine. The weather, yeah. So I'm not really going to say any content because there may be people out, but I'll just start talking anyway. So first of all, thanks for hanging out until the last day of this awesome summer school. It sounds like it's been amazing so far. And I also feel super, super lucky to be going after Noah, because I thought that was an incredible talk. I should say my daughter is in the back. She's 9 years old, so that's why you heard these laughter during his talk at various points, because she's watching a movie. And I have 4 kids, and she has this really interesting phenotype. And she's had it since she was born, where when she gets really excited about something, I don't know if she's listening, when she gets really excited about something, she kind of shakes. And it's none of my other kids do it. I have no idea why it happens, but that's why you hear her emoting a lot, because there's a lot of stuff going on when she's excited. But I'd really like to map that phenotype. I think it's a really exciting phenotype. And I've known other people who do this too. She calls it her birdie wing sometimes. So known other kids that had this. Apparently it's supposed to grow out of it, but I don't know if that has happened. And she's 9, so we'll see. Was that 2 minutes? Should we do this? That was 1 minute. Oh, I talk fast. When we're meant to talk about anything. Oh, the phenotypes of yeah, that's a good point. Let me think. That's hard. What other? I mean, hers is the most sort of obvious phenotype. I mean, the other daughter is like this crazy. My 2 younger kids are like awesome dancers, right? They just get up and they can dance to anything, like way better dancers than I am. I have no dancing skills. Let's see. Okay, thanks. I'm not going to dance. Don't worry about it. Well done, well done. Okay, I tend to talk fast, so I would love this to be interactive, and especially if you want to bring up ideas in the context of the thoughts that Noah had this morning. I would be really excited to think about this. So this is not linear regression. These are going to be latent variable models, linear latent variable models, but I'd be really excited to think about them through the lens of ascertainment, colliders, et cetera. So definitely bring that up if you have thoughts, and slow me down if you have questions, whenever. Okay, so today I'm going to be talking about something slightly different than I had on the agenda, but I changed the title yesterday. So I'm going to be talking about recent work, actually it's sort of over the last 3 years, of intersecting pathology image and gene expression data. And we're going to do this again with these dimension reduction linear models. And this is joint work with, this is driven by Jordan Ash, Daniel Monroe at Princeton University, and recently we brought in Greg Darnell, who was a student, an undergrad student with Eleazar here many years ago, but now he's a grad student in my group. Okay, so histological images are these new type of phenotype that we're really starting to be able to play with and explore. So if you think about what these images represent, these are all different histological images. We have sort of a picture's worth 1,000 quantitative traits, right? You can think about all of the differences, quantifying all the differences in these different histological images of different tissues, and think about each of those feature differences as phenotypes. So when we think about actually using these in biomedical studies, histological images are often used, for example, for cancer diagnosis, to understand tumor stage, to study sort of the boundaries of a tumor, et cetera. But we can do a lot more with that, right? We can think about how different features in the images are correlated with genotype. So thinking about population variation in these different features. Gene expression levels, which is basically what we're going to focus on today. Thinking about this in terms of cell type, so we can look at the differences in cell type based on these images. And also thinking about bigger picture issues, which is really what my sort of longer term goal is, thinking about the relationship among different cells in tissues and how these interact. So those are the sort of bigger picture ideas, but really what we're interested in today is thinking about images as a high dimensional quantitative phenotype, okay? So when we think about actually getting those numbers, right, the features that we want from each of these images, this is difficult, right? So we'd like to actually characterize, quantify morphology in these images. This is difficult to do. So, the first thing you could think about is have a pathologist annotate all these images. Obviously, that's completely infeasible. I'm going to tell you about a data set where we have over 2,000 images, and no pathologist is going to do that for us. But even more confusingly, if you have 5 pathologists annotate a single histology image, they will do it 5 different ways. Okay? So there's going to be very, very different types of annotations. There's a lot of error there. We have methods for image segmentation, and we're exploring some of those, but in general, the ones that we have available for us right now are fairly naive and do things like cell counting, measurement of cell walls, cell shape, these sort of things, and they're not necessarily the things that we think may be driven by genotype, for example. And so they're not exactly comprehensive. They're not exactly what we need. So what we're going to do here is start out with an unsupervised deep learning approach to extract features from these images. And then what we're going to do in sort of the next iteration of this work is actually change this to a supervised deep learning method. That's work that's going on in my lab right now. I'm not going to talk about that, though. OK, so the way that we actually extract these features from these pathology images is using something called a convolutional autoencoder. So I saw that Sriram and others have been talking a little bit about deep learning here. I'm going to give you a sort of 2-minute overview of what we actually use, the architecture of what we use here. So what we do in this convolutional autoencoder is this sort of deep learning approach, neural network, where we start out with a patch from a histological image. And actually, it's 128 by 128 pixels by 3. The 3 are red, green, blue. So we sort out by the different pixel colors. We put it through a number of different convolutional filters. We have this bottleneck where we have, for each of these patches, 1,024 numbers. Okay? And the idea is that we're going to learn the weights, in the encoder and the decoder, such that we reconstruct this patch as perfectly as possible with as little error as possible. Okay? So this is called an autoencoder. And then the idea is that this 1,024 feature set is going to sort of capture a low-dimensional set of features representing these images that, again, allow us to reconstruct the image as best as possible. Okay? So again, our objective here, the objective function for an autoencoder is to minimize the L2 distance between the pixels in the input and the output. Okay?
Can you give us a little bit of a sense of, so a feature here means what? The pixels with a given color, are these estimated features combinations?
Yes.
Sort of a new basis to this filter?
That's exactly right. Right, exactly. So basically, when you think about the convolutional filters, you're going to be pulling in very small patches of this 128 by 128 by 3, and you're going to keep on compressing them until you get these 1,024.
The neighboring ones.
Exactly, that's right. Yeah, you keep pulling in the neighboring ones. That's right. Until you get the 1,024 set. So the actual features, as I'm going to show you, are going to be some function of this vector right here. Yeah. This is implemented in Keras. This is an interface to TensorFlow. The code is either available last night online or this morning. Now, the question is, when we have these features, what do they actually represent? And how can we think about what they represent relative to the correlations between these features and things like gene expression and genotype? OK, so to get a little bit more detailed into this, the first thing we do for any histological image is we take the 512 by 512 segment of that image with as little white space as possible. And then we do tiling of 128 by 128 segments throughout the entire image. We actually get P of them. P is on the order of 100. We do rotation and translation just to make sure we throw a little variance in there. And then we push it through our autoencoder P times. And so what we do to actually get the features for a specific image is actually just take the empirical mean of each of those 1,024 features in this bottleneck. Okay? This is a little bit scary just because we're not entirely sure that if we actually have these values of y, the set of features for an image, that we could reconstruct a patch that looks like it actually came from this image, this manifold. but we can show you, I have a slide on what that looks like in the auxiliary. But also, the good news about this is that they're generally on the manifold. These are generally very Gaussian. So we looked at the distribution of these variables, and again, they look very nicely Gaussian. Yeah? Because it's gigantic. So it takes a really long time to train these networks, and we'd rather do it with patches. And so that's how we do it. Yeah. OK. So now we have the feature images from each of the feature images. So we can encode those feature images in this Y1 matrix, where we have 1,024 features, n samples, OK, from our n pathology images. Now we're going to use what is called sparse canonical correlation analysis, or CCA, or sparse CCA. And how this works is we're going to think about now 20,000 gene expression levels, okay? And we're going to think about those gene expression levels on the exact same n samples, okay? And I'll show you 3 data sets where we actually have data like this, but more are coming. And so the idea behind sparse CCA is that we're going to project Y1, the matrix Y1 representing the feature, the image features, and Y2 representing gene expression levels down to the shared linear subspace, X, okay? So X here is actually the exact same matrix, okay? We're going to do this by weighting Y1 and Y2, each of the features in Y1 and Y2 differently. So we're going to weight Y1 by lambda 1 and Y2 by lambda 2. And I should say that there's going to be some kind of sparse prior on lambda 1 and lambda 2. So we end up getting subsets of genes and subsets of image features that we find map onto the exact same low-dimensional component. Okay? So this is really nice for us. Now we can essentially think about clusters, groups of image features, and groups of genes that are well correlated. Yeah, go ahead.
So in the previous slide, when you learn the features, so do you have the entire tiling of the thing learned at the same time?
Yes.
So, okay.
Yep. So for one, exactly, for one image feature, we take the empirical mean over these p different segments, windows. Does that make sense?
Would it make sense to compress also the gene expressions some? They are an order of magnitude different, the sizes of the X and the Y, of the Y1 and the Y2, I'm sorry.
Yes.
In your CCA.
It would. What we actually end up seeing is that with a sort of a flexible sparsity prior, the differences actually don't end up, they end up sort of automatically rescaling.
Okay.
Yeah. Yeah, I can talk to you more about that offline, but absolutely, yeah. Does that answer your question too?
Yeah.
Yeah. So we have for every histological image, we have exactly one set of Y1 variable or YI variables, Y1 vector. That's 1,024, and based on these p different patches from here. Yeah. Okay. Okay. Yeah.
These are groups of genes that come out from sparsifying Y2.
Exactly. So groups of genes. So if you look at the
Patches.
yes, exactly. So so let's talk about this. So when we think about actually interpreting this low-dimensional space that we get, right? So what we're going to look at in the next set of slides is results that represent a subset of genes. So all the white here is 0. So we're going to think about subsets of genes and subsets of image features that we find are correlated. We find describe the variation that's common to these two different observations of the data. Okay? Is that clear? Yeah.
What's the mode of your target finding the projections from the 1 and the 2 in order to get the low-dimensional and k-dimensional projection?
Yeah.
Do you require that the 1,024 and the 20,000 features will map to the exact set or to similar points for shared features? Or?
Yeah, exactly. So essentially, so you can think about it in terms of, so we're thinking about this in terms of likelihood, right? We want to identify this latent space X where the likelihood of this dimension reduction is greatest, assuming our sparsity priors are penalizing this likelihood, right?
What makes one projection better than the other?
Oh, okay. So I should add too that we have isotropic Gaussian noise, right? So you can think about describing Y2 hat and Y1 hat as the relationship, the linear combination of lambda 1 and X, and similarly estimating Y2 by the linear combination of lambda 2 and X, right? and that you can consider that as, and then describe the differences in terms of the Gaussian noise, right?
It seems that you do y1 and y2, this projection completely independent of each other. So why
No.
would you get the projection to the
Oh,
same
no, no,
place?
no, no. You're estimating x jointly, right? You're estimating x jointly. So if you think about this, so these I would consider your latent variables, your parameters, and these are going to be your latent variables, and you can think about it in terms of expectation maximization, where you go back and forth and you iteratively estimate x jointly, and then the lambda 1, lambda 2 separately. Yep. Yeah. And I should say, actually, the method that we use to do this is from Daniela Witten, and she does this in a greedy way, where she pulls out one of these x components at a time, and then conditions on that to get the next set in a sparse context. Okay. So again, so how we're going to interpret this once we fit it is thinking about, for a specific component, the non-zero features of the image features and the non-zero genes associated with each component. And that's how we're going to try and interpret what that component means in terms of the relationship. When we look at the x, right, we have to somehow think about what these image features mean, and they're sort of, they're not very interpretable. So what we're going to do is look at the x associated with a particular component, component k. And what we're going to do is plot on this axis, these are n samples, right? We're going to plot the extreme values, the highest and the lowest values with respect to these pathology images. Okay, so to figure out if you can sort of eyeball these images, and you'll see that in some cases it makes a lot of sense, to see what sort of types of phenotypes, image phenotypes we're finding represented by the sparse set of image features. Okay? Yeah, the original image, exactly. So again, we're eyeballing it. It's very, very, very hard to go backwards and try and generate the sort of canonical image. So that's why we're doing the eyeball version. Okay. So let's actually look at 3 different applications of this method we're calling image CCA. So we have 3 data sets that I sort of mentioned before. We have 2 from the Cancer Genome Atlas. So there's one of the breast invasive carcinoma study. And in this, I'm going to tell you about one result that we get where we find that we can associate gene expression levels with a tissue feature representing sort of extracellular matrix. We have another TCGA data set from the brain lower grade glioma. And here we think about this in the context of image segmentation and cell type heterogeneity. And then we have a larger data set, the GTEx data set, where we have now 2,100 pathological images, histopathological images. And we're going to think about, in this context, genetic variants that are actually associated with tissue morphology. So we're going to talk about all these things. So, yeah.
So images can be all sorts of different things.
Yes.
Right? And you're doing an unsupervised
Yes.
compression, so it's not really linked to what you're trying to associate them to.
Exactly.
But do you do some kind of pre-processing of the images, of normalization of the images at the outset, to at least make sure that it makes some sense, right? That at least that they're all rotated the right way relative to each other, that they all represent the same amount of fish, the same surface,
Yes.
the same, some kind of normalization that is needed to make them usefully comparable.
So that's a great question. The answer is no, we don't. But the next iteration of this work in my group is focusing a lot on that. But so I should say, when we do when we pull the image features out, we do rotate the images arbitrarily, randomly, to be able to allow any kind of rotation. But the scale is absolutely critical, and you're right that that's sort of a fundamental thing going on here.
It's a little bit like an issue of aligning the images.
That's right. That's right. Aligning the scale, and we haven't done that. What we do have for some of these data sets, although actually not for the GTEx data set, is often you have a little scale parameter in the image saying, how many microns, that much is, kind of thing. And it would be really nice to be able to rescale the images so that they're all on the exact same scale. But we don't have that for our biggest data sets, so we're kind of in trouble about that. But yes, absolutely, we're thinking about those things. Yeah.
I was wondering if it's computationally outrageous to try to just maximize the canonical correlation as your feature, like as the thing you're talking about.
That's exactly what we're doing right now. Yep, exactly. So it's a little bit complicated because you need a generative model for feature extraction. We're just using variational autoencoders as opposed to convolutional autoencoders, but that's exactly what we're doing right now. Yeah, and we can talk about the complications of that too, because there are some complications. But yeah, it seems to be working beautifully. And actually, to sort of give you another sort of thing, that can scale arbitrarily large, whereas actually using a graphical model like this is quite difficult, but we have variational approximations. Okay, so I'm going to tell you about these 3 data sets. Okay, so the first one is the BRCA data set. We have about 1,500 histological images. Most of them are primary tumor. We have a few metastatic and a few normal tissue samples. These are breast tissues, so they have a lot of adipose in them, or fat cells. So we have about 1,100 tissue biopsy samples, and from this we have about 20,000 genes using RNA sequencing data. These represent about a little over 1,000 breast cancer patients. And here we actually we can think about it in the unsupervised sense, where we use an autoencoder, we try and reconstruct the image, or we can think about it in terms of the supervised sense, where we can actually estimate these images, where we can try and classify the label, where the label here is going to be whether it's a primary, a metastatic, or normal sample, okay? But I'm only going to tell you the data for the completely unsupervised version of this. Okay, so here's one result that I sort of think nicely shows how this works for the BRCA data set. So what I'm showing you here is 3 samples taken from the highest value of X. Okay, so these have the largest X value in terms of the latent component. So I'm not going to tell you about the exact features. The features actually don't matter because we have no idea what they mean. But if you sort of eyeball this set of images, and here's the set of images from the low set of features, what you can see is that the dark black spots here, the dark purple spots here, represent cell nuclei. Okay. And you can see here that there's many fewer cell nuclei, that there's a lot more intracellular connectivity here and fewer cells. And what we see if we look at the top Go terms of the non-zero genes associated with this set of this component, we see that the terms involve cell adhesion, proteinaceous extracellular matrix, ion channel binding, collagen binding. So it's related to sort of the stickiness of these cells, how these cells are sticking together, and sort of nicely describes what you can sort of visually see from these histological images. Okay? Okay, so the next data set, I'm just giving you the vignettes because I know I have to finish pretty soon. I talk slow or fast. Okay, so the next one is the brain lower grade glioma data set. We have many fewer images, about 500, and we have 401 tissue biopsy samples. And so these are glioma, so they're going to be brain type tissues. So the second component that we have, you can see here's the low and high set, or 3 images from the low and high values of X. The non-zero genes associated with this particular component are involved in synaptic transmission, synaptic signaling, transsynaptic signaling, cell-cell signaling. And again, you can see here that the extracellular matrix here is very sparse. You see a lot of nuclei with not a lot in between each of these cells, whereas here it's quite dense. And it's really interesting to see again that the genes that we're finding are strongly associated with signaling between the cells. And so that's very satisfying to see. Here's another one that I think is just fascinating in its own right, which is if we look at this, I think it's the 9th component. So these are ordered, I should say, because this is a greedy version of CCA. So this represents sort of the 9th greedy version of the proportion of variance explained. So here are the low and high values of the latent component in terms of the samples that we have. And if you look at the genes that are non-zero in this component, they are highly involved in immune response processes, defense response. This sort of blew our mind when we first saw it. We have no idea what's going on in brain. We looked at some of these particular non-zero genes and found that they were only expressed in whole blood. Okay? And then we sort of went back to this, and if you look at what's going on here, it turns out that looking at these cells, there's a much higher proportion of whole blood in these samples, in these pathology images, and a much lower proportion here. So what we're actually doing is finding cell type heterogeneity. This is the proportion of blood cells that are identified in these brain tissue samples. which is super exciting. Yeah.
So in canonical correlations, you have the first direction, which is the one of highest correlation.
Yeah.
The second one that is the second highest correlation, and so on and so forth.
Well, it's not correlation. It's going to be proportion of shared variance explained in the observations. But yes, same idea.
Canonical
Yeah.
correlation.
Yeah. That's right. Good.
Okay. You have numbers associated with this that, like in PCA, tell you how strong is that the first signal, the second signal,
That's right.
etc. So you showed 2 first signals and then a 9th signal.
Yes.
How do these things normally decay? Do you get a few very, very strong ones and then very quickly it goes down, or is it kind of slow?
Yeah, that's a great question. You know, I don't know the answer to that. We haven't plotted that. That would be a really interesting thing to plot. But looking at these numbers here, right, and also looking at the set of genes that we find that are again almost entirely uniquely expressed in whole blood. I don't know what proportion of variance this explains in these joint images, but the signal is clearly there, right? Yeah. I agree, though. I think that'd be super interesting.
Just to scale it with the other signals.
Yes, absolutely. I totally agree. Yep. Okay, so another thing we can do, because we do have the sort of a supervised version of this, is we can take now patches of these images with our classifier and slide these patches over the signal, and we can do essentially image segmentation. So here we've taken a handful of these LGG samples, slid our classifier for cancer versus non-cancerous patch over the entire image, and built a heat map. And the darker colors in these heat maps represent the points where we would classify this as a cancer tumor with more likelihood. So here again, we can effectively segment these images to identify the regions of the image where we see cancer cells versus healthy brain cells. Okay? So that's something we get out of this sort of as a side effect. This was easy. Okay, so the final data set that I'm going to tell you about is the GTEx Consortium V6 data. We have about 552 individuals in these data, about 2/3 male, 1/3 female. We have about 42 different tissues. There's actually 29 different tissues that are represented in the pathology images. So we have about like 2,200 histological images, 29 different tissue types. We have TPM values for about 20,000 genes again from RNA sequencing. And here we can think about, in the supervised setting, using labels that actually represent one of these 29 tissue types. Is anyone a pathologist in this room? Does anyone know what these tissues are? These are sort of canonical versions of 3 different tissues. Can anyone guess what this one is? Audience participation. Come on, shout it out. Looks like bacon. Ham. What is it? Muscle. What's this one?
Liver.
That's what I would have said too, but you know, this is heart. It's fairly similar to muscle. It's going to be a lot of muscle. What about this one? This should be obvious. Brain. Good, great. What's that? Yeah, heart with no bacon, exactly. Okay, so we did this sort of obvious and fun and happy thing, which was take all of the features that we have for each of these images and project them down to 2 dimensions using t-SNE, which is probably something you've seen in this class. So really the take-home point from this is that muscle is quite different than everything else. Okay, we see brain over here. You can sort of basically see that, yeah, there's a sort of bunch of all these tissues where muscle and brain kind of separate out more than everything else. I don't know what else you get from t-SNE, but someone can tell me about that. Okay, so the first component that we see in GTEx is a set of genes that identify muscle tissues. So here are the genes, the samples that are high with respect to this component, and here are the ones that are low. And we can see that we're picking out muscle samples very, very easily. The Go terms that we find associated with the genes that are non-zero with this component are all involved in contractile muscle system processes, actin binding, all things that are, most of these genes are, again, uniquely expressed in muscle tissues. And so we're just finding the identity of muscle tissues here. And this makes sense with respect to the t-SNE plot that we just saw, where muscle tissues are just sort of fundamentally different than the rest of the samples. So the next component that we looked at, we actually find neuronal tissues. So here we see a bunch of brain samples. And actually the other extreme, interestingly enough, is again muscle. And again, the t-SNE plot sort of showed you that in terms of the most different from brain is going to be muscle. So here the top Go terms are associated with synaptic transmission, other things, and most of these genes, again, were uniquely found, uniquely expressed in different types of brain tissues. So, you know, we tried a million things to try and interpret what the convolutional filters represented. Here's our 4 different layers of convolution. And I don't know how many convolutional filters you're used to looking at, but I find this very difficult to interpret. So the top level is actually something like blobs, right? So we see a bunch of blob filters where we can pick out essentially cells. The next layer is sort of my favorite, if you can have a favorite layer of a convolutional autoencoder. But you can see here that there's the filters represent essentially the density of the neurons, right? So you can see that there's going to be different types of pattern and density and closeness of the neurons. The problem with this is, as you actually mentioned before, I think there could be sort of a scale issue describing L2. So I'm a little bit concerned about that, but I think in general it works nicely. So here, L3, we can sort of see, you know, contrasted white versus black kind of things. L4 is more complex patterns that I really can't eyeball appropriately. But again, if we're thinking about the sets of features that we find, you can think about sort of combinations of these different patterns. Okay? So one thing that I generated in response to a recent talk, I think this is an appropriate thing to generate from Rafa Irizarry, was thinking about how these CCA components correlate with the known covariates of this data set. So obviously they're going to be related. And actually the reason we use CCA and not something like linear regression is because we have no idea which latent components or which observed components are going to affect pathology image features, right? We just don't know. So when we look at this, it actually turns out that a bulk of these CCA components are associated with ischemic time, time from sample collection and death. So all of these people have been dead less than 24 hours, but it turns out that time from death actually highly influences what the images look like. The 24th component here is the only one associated with this particular covariate, but is associated very strongly. You guys want to hear what that one is? Oh, it's crazy. It's cocaine. We have no idea what the genes are associated with that, but we're looking at them right now to figure out what's going on. But the fact that whether a donor with using cocaine affects images is just super interesting, right? Okay, we're looking at that now.
Between death and the moment of collection, that could also influence gene expression.
Yeah,
Like that, right?
exactly. So this is going to be gene expression too. That's right. Yeah, so I'm interested also in whether these are, you know, brain-specific genes, for example, which could be quite interesting. Okay, I'll tell you tomorrow. Okay, so then what we did was we took the CCA components. So we have, remember, a bunch of non-zero genes. We actually, since we have genotype for GTEx, we looked at the EQTLs associated with those non-zero genes, and then we looked for association between those EQTLs and the pathology images. Okay? So we have a very, very, very low-dimensional association mapping where we have a handful of EQTLs that we know affect genes that we know affect some tissue morphology. Maybe in a causal way, but I'm just going to say correlation right now. We know the genotypes will affect these two things. So we found 2 associations that I'm going to tell you about. We actually found more like 63 at a reasonable FDR. This is not a reasonable FDR, but I'm going to explain why it's not. It's a beautiful story anyway. Okay. So I'm showing you here a box plot. This is going to be the genotype on the x-axis. It's going to be the image feature on the y-axis. I'm going way over time. I hope that's okay. Okay, tell me if I should stop. And we have it's the exact same data points over here, except I've scattered them and colored them by the tissue that we're looking at. There's the FDR for this is not very low, but it's, you know, low enough that we're going to look at it. And you can see that if you look at the images here in this scatter plot, sort of there are some outliers. The association here is going to be driven by some set of outliers. And actually the outliers here are generally in muscle. And we're going to look at the gene that this EQTL came from, and this is going to be something called SYPL2. It's involved in communication between some things I can't pronounce. But I'm going to keep telling you about that gene. Hold on one second. So this is not actually the right way to do this association if you've been listening to what GTEx data has. So for every individual, for every genotype that I have, I'm actually going to have a handful of pathology images, right? So I'm sort of confounding the image features with the tissues themselves. I'm sort of mushing it all together, and that's not the right way to do it. So the way we sort of teased this apart a little bit is we looked a little bit closer at this SYPL2 gene. We saw that it was only expressed in skeletal muscle, okay? So if you look again at the extremes of this image with respect to the component that we're looking at, you can see that these are mostly muscle cells. And these are sort of muscle cells, but with funny traits. They look different, right? I mean, this looks like a sort of Christmas tree thing going on here. These are very small, thin muscle cells, as opposed to these, which look like bacon. Okay. So when we subset the images now to only muscle, this p-value gets much more intense. We see a clear relationship between the genotype and the image feature that we're talking about. And again, we can look at the extremes of this association with respect to muscle, and we see that there's obviously morphological differences going on there. So this is really nice. Again, what we're doing here is we found this genotype that we think drives population scale variation in muscle. We need to follow this up in terms of thinking about knockdowns of this gene and the relationship between muscle disease. Okay? So the next one I want to tell you about is slightly lower FDR, but we have another genotype that's associated with a set of image features. And again, there's this problem where we're doing the association testing with all of the different tissues, just because that's where we don't have a bulk of samples for a single tissue. So we don't really see a nice correlation or a relationship here, but then again, we can look at this particular gene that appears to be mediating this association, and it's mostly expressed in brain and pituitary over here. So we're actually going to look at only the cerebral cortex and the cerebellum. Again, if we look at the extremes with respect to this genotype, we're going to see a lot of brain cells and morphology difference. So here's very regular, very dense brain cells, whereas here is spotty, heterogeneous sized brain cells. Okay, so you can see differences there. So if we do this for again, only cerebellum and cerebral cortex, we see a very strong correlation between the genotype and the image features. You can even see it on this subsetted data set. And then when we went back and looked at the protein cell atlas, we found that this gene was only expressed in neuronal and neuropil cells, right on the cell membrane. And again, this tells a really nice story because now we can sort of start to understand the effect of this particular gene, or gene expression level, on the different image phenotypes that we have. Okay? So in conclusion, what we did in this study was automatically extracted features from histology images, correlate those image features with high-dimensional gene expression data. We found that the components captured cell type, tissue type, cell type heterogeneity, and different morphological features of the image data. And then we used those latent components to identify genetic variants that we found associated with population variants in muscle and brain tissue. And I'd like to acknowledge all the people who worked on this, Jordan, Daniel, and Greg, and Cheyenne Mukherjee, who I work with at Duke on CCA type methods, and all of the GTEx people who have helped me out with analyzing that work. Thank you. Questions? Yeah. Since again this is a greedy CCA, we just take the first 100 and we analyze sort of as many as appear to be meaningful. That's a bad answer, but the real thing to do, which we're doing in this sort of next iteration, is to have an adaptive number of components. And we're going to do that by sort of doing this trick that we played in methods that we developed in my group before, which is add sparsity. And as soon as you sort of see that the components are entirely sparse, in other words, all zero, you truncate at that point.
Go ahead, sorry.
Yeah.
The sparsity that you choose is another method that's very quickly, because they have this consolidation. Do you use the consolidation or you
We did not use cross-validation. So we were talking about 2,000 samples and 22,000 features. So we didn't have the signal to do that, or the number of samples. Yeah.
So as one control, I guess something you could do, and I'm curious if you've done it, is permute the columns of the expression matrices.
Yes.
So that they're not actually correctly mapping to each other.
Yes.
To see what comes out.
Yes, that's exactly what we did. So I don't think I have that image here, but we did exactly that. So for the images, we actually left them the same. For the gene expression values, we permuted the labels. And according to a number of different things, you can think about cosine distance, mutual information, whatever you want, the components basically found zero signal. That's exactly right. Yep. Yeah.
If your goal is to find correlations between the features of the images to some genes, why do you need this dimensionality reduction tree? Can't you just put the correlations directly?
Yeah, so the reason is because of this figure that I showed before. So other groups, just for the record, have been doing a lot of just association analysis, but what we found again is that there's going to be a bunch of confounders, right, having to do with time to death, whether you're on cocaine or not, and we need to be able to control for those. And so here we actually get a really nice representation. So to say it another way, if now I look at one of these features, I can first of all assume that when you think about the correlations in this feature, you've controlled, given that this is a greedy method, for all of the confounding due to all of the previously identified features, right? So you think about it, I think about each of these components in terms of the residual of the original observations with respect to all of the other components, right? So you've controlled for a lot of confounding. And the problem is when you just look at linear regression, sort of single gene, single feature associations, you lose so much of your power by not aggregating over sets of genes, which is what this method does, sets of features, also this, and by adding too many covariates, which Noah talked about earlier. Yeah.
So you're like greedily doing the CCA thing,
Yeah.
And controlling for previous components.
Yeah.
Weren't you like looking a lot at the first couple of components for like biological interpretability?
Oh yeah, so actually that was the interesting thing, right? So we find a lot of really interesting signal in later components. So the cell type heterogeneity one, where we found a lot of the whole blood genes in these brain samples, that was the 9th component. So that was pretty far down the list. Here again, cocaine is associated with the 24th component. So we do find biological signal fairly far out. And I think this is a really interesting question sort of more generally. These methods, sparsity gives you some guarantees, but these methods are often not identifiable up to rotation. So if you think about any one of these components, right, you can think about them in terms of representing a set of different covariates that are responsible for creating variation in the observations, right? Not just one. So that's why you see, again, a lot of these are associated with a bunch of different covariates, similarly. We do have the guarantee that all of these are orthogonal because of the greedy approach, right? So you essentially regress out all of the previous components and sort of effectively do projection pursuit, pulling out the next ones, the next ones, the next ones. Yes, so you can see that you split each of these confounders representation in a bunch of different components. I don't know if that answers the question, but I guess what I'm saying is biological signal can be found everywhere, and often it's spread out among a lot of different components. Yeah.
You could still sort of, again, as you were saying, you have a sparseness measurement that you can use to decide how many
Yes.
are important.
Yes.
And you have a strength of signal.
Yes.
Share of variability of covariation explained that you can use
Exactly.
to see if it makes an elbow. But that said, it's true that interesting signal can
Yes.
be way down.
Way down in the tails. Yep.
In terms of trying to interpret these things, you were making the point before that it can be very, very difficult. So this is a way to relate them to potential confounders.
Yes.
But in this actually, I remember a similar exercise done in a completely different context a long time ago, when people ask you to explain,
Yeah.
right? And what is this first component? Who knows?
Yeah.
You can start essentially regressing it,
Yeah.
correlating it
Yeah.
There's all sorts of different things as
That's
long
right.
as you have them measured on the data.
That's right.
And sometimes that's actually an indirect way to find out what it means.
That's right.
In addition to this, for instance, can you do it with some sort of canonical, it's not the
Yeah,
right
yeah.
word, but sort of typical features of imaging?
Yes. Yes. So if we do, so yes, that's another direction we're going right now, like I mentioned before. So we're doing sort of using all the possible histological image segmentation methods that we possibly have, annotation, and seeing if any of the features that we extract here are correlated with sort of cell count or something like that.
Yes.
Exactly. Yep. So I think it's a good way, but again, I am extremely nervous about over-interpreting these components from latent variable models from exploratory data analysis because of this rotation invariance. Yeah. Good. Okay. Thanks.